Hello, everybody. Oh, you got it back, Phantom? Okay, good. You did not message me to my knowledge, but I'm glad you got it back. So it is safe if you message me now. Is that right, Phantom? I'm on the right track. My goodness. And also good morning, or afternoon, or evening, or whatever time it is where you are. It is morning for me, everyone. I know, a rare Jihei morning stream. You're going to see more weekend morning streams happening. Poor Astral is going to have to deal with weekend streams. I have no choice, okay? I have a day job. What am I supposed to do? What am I supposed to do, my guys? I have no choices. I have no choices. Today is an education stream, so have yourself a pen, paper, or device at the hand. There'll be some little activities for you to do to check your understanding as we go through. And just a reminder that the reason for these streams is, I know a lot of VTubers have talked a lot about how they feel like they didn't get value out of school, or there's things they wish they'd learned about, some of which I'm quite shocked they didn't learn about in school. Like, I'm learning that there's some curriculums in the world that distinctly unimpress me. All curriculums are flawed. They have to make decisions, they have to prioritize things, but there's some curriculums where I'm not really sure what's left in them. So I'll often hear things like, I should have been taught about XYZ real life skill in school, for example, and the curriculum I teach in does in fact require you to learn those real life skills in school. Although I do disagree somewhat, that's not really what school is for. Your parents should be teaching you that. Schools are picking up the slack because of terrible parenting. But even in terms of academic study, lots of very basic principles that I'm told I didn't learn at school because I was allowed to drop that subject at an incredibly young age. And I understand wanting to curate your education system so that children aren't overwhelmed. There's definitely some things I think we're all wishing some adults had learned in school now. I'm looking at you, global leaders. Astral, is my sound level okay? Because I don't really know what's going on with my mic, because I never change it, and sometimes you tell me it's too loud or too quiet. Crazy. Today we're going to be talking about the characteristics of living organisms and how we classify living organisms. And we're going to teach you all the way from what you would learn at 11 years old through to, give or take, 18 years old. This is a pretty low-level module, so although there are A-level stuff, 18-year-old stuff, it's not going to be a challenging one for most people. There will be some other topics that are going to go much more deep at that level. This topic does get a lot more deep at degree level, but we're not doing degree level yet, at least until we've finished all of the streams up to 18. Let's get our baseline, our foundations. You can't build a house on a bad foundation. This is a really long presentation, so I'm going to start. We're going to have some regular breaks built in, because you guys need breaks in your learning too. I wouldn't make a student do lessons this long, so why would I make you guys voluntarily come here and sit with me for so long? So, what are we going to be talking about today? We're going to be talking about living organisms. Now, living organisms can be animals, plants, fungi, prokaryotes, or protoctists, okay? And we're going to talk a bit more about what all those things mean later. And we're actually going to go into some more detail with a couple of them. And they all share, all of these things, everything that belongs in these categories shares a set of common characteristics. And we can remember them using an acronym, Mrs. Gren, okay? Mrs. Gren is the 7 that you would learn in lower school usually. There's sometimes curriculums will teach Mrs. C. Gren or Mrs. Grenk. That is because control has been added. It's a bit controversial because an argument is that these things are the things you control, and control's too vague, but sometimes you will see it. So if your curriculum asks you to include control, if your curriculum asks for 8, then that's what you need to provide. But if you know Mrs. Gren, you pretty much have a solid basis. And you can see MRSGREN, and there's some little pictures. Take a look. Can you guess what any of Mrs. Gren are? Are you able to have a little guess? A little guessy guess? No? Yes? How are we feeling? Mrs. Gray. Some of them are really obvious, and some of them, honestly, not so much. Some of them definitely catch people out. I definitely have students who struggle to remember them from time to time because there's just so many different ones. Okay. So first of all, M, easy. Movement. Movement. Now movement, so all organisms move, and a lot of people, they miss out plants as they don't move. But if you put a flower and you put it on a windowsill, it's going to over time be leaning towards that sun, and if you were to turn it, it will start to lean back the other way. It's a very small amount of movement, but it is movement. Animals, we're all pretty comfortable they move, I'm assuming, for the most part. Now obviously sometimes students will take this too literally. What about if it's disabled, etc., etc.? This is the capability of the species, not necessarily of individuals in the species exhibiting variance. And we'll get to what a species is later on. And then there's 2 R's. So it doesn't matter which way around you put them, it's just an acronym to remember them, but I've done respiration first. This is chemical respiration. In general, in high school kind of level science, when we're talking about respiration, we mean chemical respiration. We don't use respiration to refer to any part of the ventilation process at high school. So we mean chemical respiration. That's the reaction inside your cells that breaks down the food we eat, the nutrients in the food we eat, to release energy for our metabolism, for the reactions happening in our body. Metabolism is the reactions happening in your body. And those chemical reactions form the basis for a lot of what we're going to learn about in the other biology topics. Okay. So the nutrients we have there, these are carbohydrates, sugars and starches. They get broken down, and when they get broken down, they don't create energy. Energy cannot be created or destroyed. They release the stored energy from the carbohydrate, and we need that. And all living things respire, even plants. Sometimes students will mix up photosynthesis and respiration. They are 2 totally separate processes, and even plants have to do respiration. What do we think S is? Let's have a little think. What could S be? This is one that gets students. They normally are fine with respiration, movement, but S is a little bit tougher. So S is sensitivity. So S is the ability to sense changes in your environment. And for animal species, we often think of our 5 senses, right? Seeing, smelling, hearing. But plants have senses too. They have the ability to detect gravity for gravitropism. That's why when you plant a seed, it knows which way to grow. It knows to send its roots downward and its leaves and shoots upward. And plants are also sensitive to sunlight, which we've already mentioned. So sensitivity. Bacteria can sometimes be sensitive to chemicals around them. They can sense the chemical that they need. And growth. All living things grow. They make an increase in their size and dry mass. Okay? So they don't increase by just absorbing water. They actually increase the mass within them. Look how cool these cell division pictures are. I know it's a bit of an aside, but as a science teacher, I love really cool pictures. And cell division pictures like this are incredible. And we are going to go into a lot more detail about cell division when we get to that topic. But just look how amazing that looks. And how incredible it is that with a microscope we can see this. Thank you, Robert Hooke. Thank you, thank you, thank you. And then the second R is reproduction. Okay, so reproduction is that any living organism can make more of the same kind of organism. It can make more of itself. There's different types of reproduction, like asexual and sexual reproduction, but all living things do make more of themselves. Okay, they do that. Which means we've got E and N left. What do we think E and N are? They're actually quite connected to each other. What do we think? Oh!
I came here to drink milk and kick ass. And I've just finished my milk.
Thank you. Thank you so much for joining. Oh my goodness. It's definitely a squad up moment. I'm not sure this was a great raid choice. We're doing an education stream. We're learning things. This is not a typical stream. But hello, Raiders. My name is Just Shihei. I am a variety streamer with a quite insane variety, so I always stick to similar days of the week. So if gaming is your thing, that is Mondays to Fridays usually, and sometimes a little bit on Sundays. Oh, was it that bad? DJ, was it that bad? And you can tell what day of the week to come and see me by looking at my stream schedule. I do stick to the same day of the week for things each week. So cozy gaming is the same day each week, things like that, just to make it a little bit easier for you guys to deal with my unhinged ADHD need for stimulation. It's oddly jank for a game. I don't know, I feel like more recent games are highly jank. But today we're doing Britesparks Hagwon. So Britesparks Hagwon is a project I have where I basically just teach you guys things that you probably learned in school or should have learned in school and have forgotten now. And you just get to relive that moment and come and join a classroom. Especially for some of you who I know have had some pretty traumatic school experiences. Just giving you a chance to learn something in an environment that doesn't make you have those negative associations. Because learning things should be fun. And by fun, I don't mean it should be gamified. I mean it should be enjoyable just to explore your curiosity. You shouldn't need to gamify it because you should enjoy the feeling of progress. And everyone can have fun. And I'm qualified to say that. Yes, E and N. We need to get to E and N in our Mrs. Gren, or Mrs. Grenk, if your curriculum's feeling edgy. So what are we doing for E and N? What are we doing? Well, E is another one that's tripped students up. The S and the E, that's the one that always gets my students every time. And E is excretion. It's the removal of waste products of metabolism and substance in excess of requirements, which is a fancy word for the obvious things like peeing and pooping. But it's also things like in plants, excreting the oxygen from photosynthesis, or in humans excreting the carbon dioxide from respiration when we breathe out, or even plants excreting some carbon dioxide. They don't really do it so much, they reuse. Urea being excreted by our kidneys is part of the excretion system, yes, so we're good with that. But sometimes people forget that the gas exchanges are also an excretion. Things that bacteria do to expel, that's also excretion. Excretion, X-E-X, like exit. Exit is the way out. Excretion is us putting things out. Okay, and in science it's always really useful to think about our prefixes and suffixes of words, so you'll hear me do that a lot, comparing. And then the last one is sort of the opposite of excretion. It's nutrition. We have to get those nutrients and materials from somewhere. Animals, it's easy for us, we ingest food. Plants, they absorb certain things from the soil and they create their own energy stores. So nutrition is not just your energy though. It's not just your carbohydrates, it's all of the materials you need for your growth and development. So in humans, that's your vitamins, that's your minerals, that's your fiber. None of you are eating enough fiber, guys. None of you. I'm telling you now. You're not eating enough fiber. And I'm going to also warn you that when you start committing to I'm going to eat more fiber, the first couple of weeks are going to be uncomfortable while your body adapts, but you need that. Colon cancer is fast becoming one of the biggest killers in the world of people under the age of 40. That is terrifying. Eat your fiber. You don't need men in supermarkets with their shirts off shouting at seed oils or telling you to eat butter sticks as they are. You need more fiber. It's boring and it's not fun, but you need it. Actually, it is fun. I like eating more fiber. Some good food with fiber in it. It's surprisingly hard to get a full amount in your diet, though. Who knew? Who knew? Nutritionists everywhere must be dying at watching these protein anti-seed oil crazies in the supermarkets. It must just make you wonder why you qualified. You don't need qualifications. You just need to sit in a car with some sad music playing, making an 8-second long TikTok with text over you. That's what you need to do. You don't need qualifications. Oh my gosh, is my face busted? No. Okay, so my model's working today. Good to know. Just doesn't believe in working for Star Rail. All right, so we have movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. I hope you have your notepad ready, or your device, or whatever it is you're bringing. Okay, if you're with someone sat watching, you can do this as a pair. If you're not, you could do it by yourself. If you want to flex it in chat, you can flex it in chat. I'm sure that someone will love copying off of you. Can you match up the words to their definitions? A lot of scientific understanding comes from not knowing what the vocabulary means, and a lot of people use scientific words to trick you. They use these words that are not part of our day-to-day speech to make you believe they know what they're talking about. You can give yourself so much power by knowing what those words mean. So much power. So I'm going to give you 2 minutes. 2 minutes of not having to listen to me yap while you have a go. All right. See, Faye, I said to everyone, I'm starting with something simple, but there are, we forget this stuff. We don't sit in school all the time. Okay, I'm going to pop some answers up. We haven't even got to the juicy stuff yet. What do we do? How do we do? Did you get it, Astral? Did you get it? Process of elimination is what I always tell my students to do in their tests, by the way. When they're like, oh, I just don't get it, so I gave up. I'm like, oh, you're giving someone else a C grade then. Okay. Because someone else will use the process of elimination, but come on. Did you get them all? Did you? You tried. That's what the learning process is about. We try. And we're starting easy. We're starting with classification of organisms because it's a nice, easy. Well, I say easy. Obviously, it's easy for me. It's easy for me, but it's easy as in it is something that people can pick up and they're likely to remember more of from school. When we get onto the harder topics, I don't want everyone being like, I haven't been in school for so long. How am I supposed to do this? You confuse yourself with your own answers. Okay. So, movement is... Oh, I've lost it now. Wait, my answers didn't come up properly. No. Okay, I'll just go through the answers and make sure then. Hang on. Okay, movement is B, so it's 1B, an action or organism causing a change of position. Do my answers come up if I click again? Let me just check. No, they don't. Oh my goodness, I broke my slide. It's fine. So 1 is B. 2 is E. Wait, oh, it did come up. Okay. Yeah. Yeah, it did. Okay, I panicked myself then for no reason. Okay, you did get them right. Nice. Good. That made sense of my weird answer key. It's good. One of the most common areas my students struggle is when this stuff seems pointless, boring, right? So they don't learn what the words mean because it isn't the most fun part of science. But then when we get onto the fun stuff, they don't have the vocabulary to understand what's happening. So let's do a little true or false. A little true or false. Astral, do all living organisms have eyes? Is that true or false? To be fair, Astral, as well, although English is your first language, you are Filipino. It's not like your native language. Do all living organisms have eyes? Do they? Do they, guys? Do they? Do they? They do not. That is false. All living organisms do not have eyes. Good job, Astral. Do all living organisms breathe? Do they all breathe? They do not. They all do respiration, but they do not all breathe. What about, do all living organisms need nutrition? Yeah, they do all need nutrition. Yes, you guys are experts. All living organisms can reproduce. Do again, produce, make more of themselves. Do all living things reproduce? They do. They don't all do sexual reproduction, but all living things do reproduce in some way. And that's going to be really important when we're talking later about something that is controversial in living organisms. What about all living organisms do digestion? This one is the meanest one up here. It's actually false. Some living organisms do just use molecules as is. Digestion is the breaking down of the molecule, and some simple organisms cannot break down molecules, they just use them as they are. Remember, another thing that trips people up is they start thinking about whether every individual in a species can do something. For example, an individual can be infertile and not able to reproduce. Okay, an individual can not necessarily reproduce. Some plants can actually digest. Oh, I'm so excited to get into this now. I love this stuff. Okay, we're not going to do this on this particular day because I think we need to move on. This is a little bit basic for us. But when you're studying things, this is also something that can come up a lot. What I am going to do is just leave this on my screen for a couple of seconds. If you're watching this as the VOD, if you're someone reviewing and studying and you want to pause it and have a go at this, you can pause it now. And if you need the answers, you can pause it now so that you can check the answers for you. This is for any of you who are using this as a VOD for any review or prep for any exams you might have. Okay, so now we've done a little bit of a review on things being alive, and we've got to keep Mrs. Gren in the back of our head for later on. It's going to come back up. I haven't just done this to be crazy, although I am a bit crazy. I haven't today. We're going to talk about classification, and we're going to then link this in later to biodiversity and conservation a little bit. But then we will talk more about biodiversity and conservation in a separate lesson, okay? Because classification itself is a huge topic. And I'm going to go through it as quickly as I can, because these are supposed to be hour-long streams, and there's no way this is going to be an hour-long stream. It's going to be way longer. So let's see if we can keep it as trim as possible. If I'm going too fast, please tell me to slow down. If you have any questions, please put them in the chat. I'm here to answer your questions. They can be a little bit off topic. I'm always letting my students go off topic. Let's stick to the science though. The fabulousness of Jihei is for another time. So let's start with the classification systems. And I've got a challenge for you, okay? It's a little bit of a challenge. You can do this just as a thought experiment. You can just think about it, or you can write it on some paper, or you can do it in your books. If you're watching this as a VOD, I really recommend you do it properly. So set yourself up now. You're going to try and group the organisms you see into categories. You can pick how you categorize the organisms, okay? But how would you organize them? How would you put them into a group? Oh, wait. Let me get it up now. This one? Oh no, that's something very different. Let's not do that. What is it? This one? Yeah. Where's it gone? It's disappeared. Is it this one? It is. Okay. Okay. So here are the organisms for you to oh no, have I broken everything? It's really embarrassing if I have, isn't it? Okay, have a go at, well, that's just crazy. Have a go at popping these into some groups. Have a little try. What do you think you can do? Have a think. Oh, this is another opportunity for me to shut up for a minute or so, isn't it? I know, streaming crime 101, but I want to let you be able to think. So I'm going to give you 2 minutes. All right, what did we have? Did anyone have any thoughts on how they did it? Anyone want to share? I'm not going to make people share, and it's really awkward, especially on the stream. But if anyone wanted to, I will give you a chance. Did we organize them by color, size? Did you organize them by number of legs? Did you organize them using a system you're already familiar with? By the time my students get to me, they've already learnt some words for classifying organisms in lower school, and they always refuse to do this task of their own creativity. They always refuse to come up with a new system because they already know one. And that's one of the perils of knowledge? Is it stopping you thinking about creating new knowledge? You can't be a good scientist about it. You organise by their code scales, but I quickly saw a flaw in that, but oh well. What was the flaw? Where did that trip you up? It's a really good way to look at organisms. We've got very little information here. We don't even have Latin names for some of them. Ah, yeah, you ended up with only one that has feathers. Yeah, yeah. Because it's quite a small sample size of organisms we have here to try and organise. Maybe that's the only one that belongs in that group. Maybe. Okay, let me switch back to our main PowerPoint now then. I will probably have to resize it because it's going to be tiny now. Okay, there we go. It's probably going to be more resizing because I picked a bad slide to resize, but we're all good. At least we're back. Okay, so the key point here is that classifying organisms is actually really hard. Okay, it's not an easy task at all. It's not something that oh gosh. Hang on a moment, guys. Let's fix this. Oh good gosh. There we go. That's much better. Thank you. It's actually really, really difficult, and scientists have not always classified organisms in the same way because we can't. As we've learnt more information, we've had to update our knowledge. We can't just assume that science stays still. And in fact, this is where a lot of people go wrong when they're trying to debunk science. They'll go, well, you won't even say this is true, or you've changed your mind about this. Yes, science takes the evidence and knowledge we have and creates explanations. As we gain more knowledge, we may need to change those explanations. And classification is a beautiful example of this. Okay, we began the same way you did, Faye. That is how most of us how most organisms were classified. We looked at what we could see. We looked at their coverings. Were they fur, feathers, scales, skin? What was covering them? We looked at whether they had legs, fins, flippers, beaks. We looked at things we can easily visually see, like do they give birth to live young? Do they have eggs? Do they produce milk? Things like that. These are physical features that we could easily classify. And we did, and still do, classify organisms predominantly based on those features. Okay? Now we're going to get later on to why that's changing, but for now, although you may have found with that sample that not super helpful, it is the basis for how we classify living things. Okay? And they can be classified into groups or based on those features that they share. So at the most specific level at the bottom, we have a species, okay? And species are going to be the most similar. Then above that is genus, family, order, class, phylum, kingdom, and we're going to talk about domains at the top. And these are, as you get higher up, your organisms will be less similar based on those physical features, all right? So just to be clear, though, about species, because sometimes species can be confusing. For example, both of these are cats, the same species, the domestic cat, Felis catus. But they look very different, okay? And dogs is even worse. And this can be true that animals can be of the same species but not look particularly similar. A species is specifically a group of organisms that can reproduce to produce fertile offspring. And the fertile offspring is important because someone will always bring up ligers and tigons. And ligers and tigons are not fertile. So when a lion and a tiger have offspring, they can have offspring together. They are fairly closely related species, but their children, the mix of their two species, are infertile. They cannot produce offspring. So a group of organisms that can reproduce and their offspring be fertile, that is a species. That's also why ligers and tigons are not species, because they can't produce fertile offspring because they are invariably infertile. And members of a species are almost identical anatomically and behaviorally. Mules are also not a species. That is correct. Mules is a great example. When I did my year 6 SATs, which is exams you do in the UK when you're about 10, 11, I think they still do them. I hope they still do them. And they test things like your reading skills. I had to read a text on mules. I had no idea what a mule was till I read that text. But yes, they can look different though. So these two cats can look very different, but anatomically they have roughly the same structure to their skeletons, their eyes work in the same way, and their behaviors will be similar. Obviously they're domesticated, so their behaviors are affected by training, but their inherent behaviors will be similar. Okay, and they are the same species, they can reproduce and their offspring be fertile. They're different breeds. So when you have things like this where we have created selective breeding, we have taken a member of a species with a particular trait and we have bred them together to enhance that trait. You're a different breed but the same species. Okay. And this is called the morphological species concept. So this is where we take species that look similar, share key morphological traits, things that are morphological, so your physical traits, and we put them together and say, are you different enough to warrant a special separate species? And this is really practical for species that no longer exist. So for species where we can't do breeding experiments or they were born before modern theory, fossils, we pretty much have to use morphological species concept. There's very little we can do unless we can extract DNA. They can be applied to extinct things. It's observable and measurable. However, there are some things that mean that at a low level this can be difficult. Scientists have to be very careful how they apply it. So there's a phenomenon in biology called sexual dimorphism, and sexual dimorphism is so common in birds. Birds are the most beautiful example. A male and a female peacock look very different. The peacock and the peahen look very different. In fact, birds, male and females of the same species, often are so different that bird breeding groups, they name birds by their gender. So peacock and peahen, for example, are simple examples. But if you look at hawks, like falcons and things, there will be special words for the male and female of the species as well, because they're just so morphologically different. Also, some species have extensive variation within their species, okay? And sometimes there can also be the other problem. There can be species that look really similar, but are actually really genetically different. And there are also subjective decisions, like which features are the most significant variation that need a new species is subjective. It is up for debate. So really, we try to stick to only using morphological species concept when there is no other data available, or using it alongside other data, okay, so that it's more accurate. Okay, it is excellent for extinct fossils. We have very few ways to put those into the structure of living organisms. Ecological species concept is different. It defines a species based on where it lives, its environment, its habitat, its use of resource, and its interactions. Okay, so each species on Earth does have environmental conditions and resources that it's adapted for, like temperature, food, habitat, and they use different resources. Okay, so it could even be different parts of the same tree, different depths of water, different types of coral that they live in. It could be that specific. So the species is distinguished by its adaptations to those challenges, okay? The anatomical features that do that. And for advantages, it really helps us compare the ecological relationships, which is really useful when we want to understand the importance of biodiversity or look at saving species or look at things like that. The problems is how do you define where a niche actually is, and that this can change over time. That's the whole point of evolution. And that also related species may join the same niche, but they may still be a different species. The ecological species concept is really something to be used alongside other methods, or to be used separately to classifying organisms, and to only classify organisms when we're looking from a biodiversity point of view, not to identify individual species. So, we're not going to work in groups of 3. I'm sorry, these are my slides I use in actual lessons, so you have to forgive me. In scenario A paleontologist discovers fossils of ancient marine reptiles and needs to determine how many different species are in the fossil bed. Okay? What are they going to be looking at? Are they going to be looking at the morphological model or the ecological model? Most likely morphological, because they're extinct. They could use ecological for the layers in the fossil bed, but it's probably going to heavily lean on morphological because they're extinct, so we can't witness their behavior. And ecological ones, we kind of need to witness their behavior going. What about scenario 2? We have a biologist looking at 2 populations of frogs that look identical, but they live in different types of wetlands. Is that going to be morphological, to do with their physical features, or ecological, to do with their relationship with their environment? So that is nailed it. They look identical. If they look identical, morphological is not going to help us a lot, but ecological is going to help because they have different mating calls, different types of wetlands they go through. It is going to be much more ecological. Potentially the mating calls could come under morphological a little bit, but it's going to be primarily ecological we're looking at. Scenario C. The geneticist is studying bacteria that reproduce asexually, and he wants to understand their diversity and evolutionary relationships. So for this one, I think there's a pretty strong argument for both. Okay? And by both, I don't just mean either. I mean, there's a pretty strong argument for using both of them. You have a lot of information available, and there's no limiting factor to using it in that one. So once we can classify animals, we can organize them into classification systems. And one of the most common classification systems you'll see uses something called the binomial system. So you know when we see those Latin names, like famously gorilla is gorilla, humans are Homo sapiens. Those are the binomial naming system. And the binomial naming system uses a system where it's the species and the genus, which is the rank above. So genus is closely related species that cannot interbreed to produce fertile offspring. But some within the same genus can interbreed for non-fertile offspring. Species are the identical ones, okay? So this Felis genus, Felis, includes wildcats, our pet cats. They can't all interbreed successfully with each other, but they are all very closely related. We've all seen the videos of like African wildcats that behave and look very similar to domestic cats now. Okay, but they can't successfully interbreed. And they do exhibit some behavioral differences too. And we call this the binomial system. So the house cat is Felis catus, and the domestic dog is Canis familiaris, yes. Actually, coyotes and wolves is the absolute best comparison, I think, to the domestic cat and the African wildcat. I think actually a domestic cat and an African wildcat could probably breed successfully, but they have enough variation in their behaviors and morphology for us to still think of them as a separate species. When humans get involved, we sort of break the natural systems a bit. And when we're writing these, you always write, I know this is like a stupid thing, but it always really bugged me at school that I actually wasn't taught this and I wanted to know. So the genus starts with a capital letter and the species starts with a lowercase letter. Okay? This is binomial nomenclature when you get into upper school. Okay? Genus and then the species name. And the species name is always lowercase. And they're always written in italics when they're typed, or if you handwrite them, you underline them, okay? Once you've written it the first time, E. coli is a great example of this. We don't expect you to write Escherichia coli every time. Once you've written it the first time, you can write E.coli, okay? You can abbreviate it. And we use this deliberately to avoid confusion. So lots and lots of common names exist for the same animals, not even across countries. Even within a culture, there might be multiple names for the same animal. And so this is like a way for scientists to know wherever in the world they're communicating, they can use this traditional name, usually Latin, but it can come from other languages too. And this name will be the same everywhere in the world. It's the same reason we use the same periodic table everywhere in the world. Yes, was it originally from Mendeleev? Yes. Does that mean that the rest of the world shouldn't adopt it? No, because we need this common language for science to be communicated. And one of the first ways you learn in school to identify these things is something called dichotomous keys. So if any of you grew up in the era of magazines, you know with these, find out who your Sex in the City bestie would be, find out who your Twilight true love is, that type of stuff. Those quizzes are all dichotomous keys, basically. You're asked a series of yes/no questions to identify a final response. And that's how dichotomous keys work. And you'll sometimes see them written like the one on the board here on the screen, but sometimes you'll see them written as kind of flowcharts, like family tree style diagrams. So question 1 gives you a choice between 2 alternatives. It doesn't have to be a yes or no, but it could just be 2 choices. Or it could be a question with a yes and no option. And then it either tells you what that animal would be, or it tells you to go to the next question until you can identify the animal based on a list of these questions. And in school level now, students are expected to be able to make these, okay? Because it encourages students to study the differences in organisms, be able to vocalize, verbalize, and explain the differences, use the vocabulary, and be able to apply that understanding into their own. It also shows an ability to follow a logic plan. So sometimes people go, oh, why do I have to learn to do this in school? It's not about the actual task itself. You might never be asked to make a dichotomous key in your life, but it's about the ability to apply the skills and understanding and the way your brain works. So, when you're coming up with a plan, is it warm or cold-blooded? We need to split them. Okay, so far we've not managed to identify any individual species, but we've started to create a tree that could. So we need another set of questions. So I could ask, does it have feathers? Yes and no on the right. And that's going to give me immediately a set of animals that I can identify. Where's my thing gone? Ah, there. Okay. I now know that if it has no feathers, it's a cow. If it has feathers, it's a bird. Okay. Is it warm or cold-blooded? Cold? No. It doesn't have limbs, it must be a fish. Okay, but the frog and the lizard still both have limbs, so I need a question to split them up. So I'm going with, does it have scales? And no is a frog, and yes is a lizard. So now I have this, I can write them in the style of a dichotomous key. So this flowchart is what you'll be used to from the magazines, but we're going to write it in a more scientific way. So all we're going to do is take our statements, write them in a numbered list, and direct ourselves to the answers. Okay. So, was it cold-blooded? We have options of answers of cold-blooded or warm-blooded. They can get remarkably large, but they are used in identification charts. So they are used on a small scale if you're trying to identify, let's say you're studying those frog populations we talked about, Faye, where there's two very similar frog populations and you need to be able to identify the two. There will be a smaller version of this key that breaks down the features you're looking at. Bacterial populations, they're still commonly used for. They're not used for wildly large animal studies, okay? They're used for verifying if you found a new species more than anything else, okay? So they're used for, you've got a selection of similar species, you're going through the questions, and if you can get a matching set, you probably don't have a new species on your hands. They're very commonly used for small samples of invertebrate populations for people doing highly specified study. But you are right, in modern day science, they are very limited in their practical application. We learn how to do them in school because they are a historical part of how we've developed our scientific understanding, and it's important we understand how we've gotten to the understanding we have today so that we don't have people going, oh, well, I just don't believe it, where's the evidence? Okay, well, here's the journey we've gone on. But also because the reason it's tested in school levels is because it tests a whole bunch of fundamental underlying skills. Okay, so the ability to critically think through the source, the ability to analyze morphological features of animals. Those skills you do still need. And this one task can test a student on all of those skills. I hope that answers your question. You could do a dichotomous key for everything. So like this lab glassware, this is all lab glassware that students need to know to be able to write methods for lab experiments, to be able to write out experiments. So has it got a pouring spout? Yes. No. Okay, if it has a pouring spout, we go to number 2. Has it got a broad base? Yes, it's a beaker. If it doesn't, it's a measuring cylinder. Measuring cylinders, beakers, they both have pouring spouts and different size bases. If it didn't have a pouring spout, we know, has it got straight sides for the whole of its length or not? So if it has straight sides for the whole of its length, it's a boiling tube. If it doesn't, it's a conical flask or a round bottom flask. How can we tell the difference of these? Sloping sides. Okay, so these types of keys are not just used within scientific animal classification, but they were how early scientists would have started doing it. And they are still relevant on small sample sizes today. So why do we classify animals though? Why bother? Other than human satisfaction about putting things into a box, it does actually serve a purpose. So they help us work out evolutionary relationships. So if we know that two species have a common ancestor, they're going to be grouped together. And if we know that they've evolved independently, we classify them separately. And this allows us to predict, potentially, relationships that we've seen. Okay, so here we go. Here is the similarities in the structures of different vertebrates that hints we had a common ancestor. So even though all of these belong to different classifying groups, they all belong under vertebrates. Vertebrates are animals with a backbone. And you can see they do all have a scapula. And the scapula is probably the most different of the bones. Then they all have a fairly similar humerus, to be honest, a long bone. And then they all have a radius and ulna, a pair of bones working together. They all have these small carpals, metacarpals, and phalanges, although again, the carpals, the metacarpals and phalanges show a lot of distinction, especially the phalanges. But this whole section in the middle, the carpals, radius, ulna, humerus, they're all very, very similar across species in the way that they work. Okay? And this gives us some clues there's probably a common ancestor involved. It's not the only piece of evidence, but it is one of a portfolio. On its own, it would prove nothing. Added into everything we have, they do. So here, when we look at the differences between the behavior of these two species, they do look kind of similar at first glance, but we spot so many differences to their actual anatomy and so many differences to their behavior that actually we think the pangolin and the armadillo do not have a common ancestor. So we would classify them differently. But we need to look into the depths of all those things we talked about, the morphological and the ecological. And morphological does not have to be the visible features. On the surface, these two do look similar. Inside, they do not. And this has been traditionally really hard because lots of animals like the pangolin and armadillo will have been misclassified because we had limited ways to study them. So what's really helped is our understanding of DNA and gene sequencing, because we can literally look at the genes. And if the genes are different, if not the alleles, so not whether you have blue or brown eyes, but whether you have those eyes, if those genes are the same, then we know you're a same species. If they're not, you're a separate species. And we can look for similar genes that match the morphology. Okay? Every species has a unique series of bases in its DNA and unique set of genes. Okay? And we have sequenced the human genome. It's called your genome, and the human genome is sequenced. Okay? It was a big thing when I was at school, the Human Genome Project. And it means that we know what genes humans have. We don't know what versions of those genes you might have. That depends on your parentage and your inheritance. But we know which genes you will have. And that is how we can tell which things are separate species. So this wolf and this husky look really similar. They look really similar, but they have different base sequences in their DNA. They have different genomes. So we can tell they are a different species when we look beneath the surface. And they're also a way for us to see which more common recent ancestor might be similar compared to decents. So people are often shocked that human DNA is actually closer to chimpanzees than orangutans. That means that we diverged from chimpanzees more recently. We're still very closely related to orangutans, a 3.1% difference in our DNA. It's nothing. But often, because orangutans have such a human look to them, and often quite human behavior to them, people assume that we must be really closely related. So alleles are when you have the same gene but different versions of it. So if you have a gene for hair color, for example, to oversimplify it, you have a gene for hair color. One allele for that might be brown hair, one allele for that might be blonde hair. This gene is still for hair color. The alleles can be different though. So having that gene for hair color makes, in that place in your DNA, makes you human. But you could have any allele of that gene, brown hair, blue hair, blonde hair, blue hair, humans? No, not genetic, just made that up. But you could have any allele of the hair color in that, yes. I mean, there is a whole topic on genes and evolution, which I obviously won't be going through today because this topic alone is a crazy length of time. But we will go into alleles, genes, chromosomes in a lot more detail. So yeah, this shocked me. I honestly thought we were more recently diverged from orangutans, but it actually means we have a common ancestor closer with the chimpanzee, even though we share so much behavioral and physical connection with an orangutan. So we can actually have a look at this. So we have orangutan, gorilla, chimpanzee, and bonobo, and human. Okay? And if we go back from human, here we meet a common ancestor for the chimpanzee and the bonobo. That means that we have the most recent common ancestor with a chimpanzee and a bonobo. If we go back again, we find our next most recent common ancestor with the gorilla, and actually we have to go all the way back to 13 million years ago to get a common ancestor with the orangutan. We are still have a common ancestor. We're still only, we're only 13 million years of evolution away. But we are much, much closer. Only 6 million years, our common ancestor with the chimpanzee and the bonobo. But remember that in between this time and this animal was still evolving. So we were evolving and losing common ancestors. And then when they branch off, they keep evolving, but in a different way. So what that would imply here is, even though we don't have a recent common ancestor, a lot of the orangutans' evolution in those 13 million years was creating a lot of adaptations we recognize. So at some point here, we continued on a similar path, the orangutan, and perhaps the chimpanzee, and even more so the bonobo, they diverged a bit from us. but that is separate them to them being related to us. Okay. We're going to come back and talk about features, but I am going to let us take a break because it has been over an hour of mostly sitting and listening to me yap. So we're going to take just a 5-minute break. Please go to the bathroom, stretch your legs, whatever you need to do before we move on to talk about the features of different types of organisms and the different types of classifications that there are. Hello, my name is Just Jihei, and you might know me from such classics as Kidnapping Filipinos, Platonic Men in My Basements, and my iconic attempts at scuff-free streams. Wow! Yes, you heard it, scuff-free streams. After many years of work, tireless effort, I have created a product that can scuff-free streams and sprays it right in your face. So if you are fed up of scuffed streams, chatters saying such things as, "Have you tried turning it off and on again? Your mic is muted. Your mic is muted. Your mic is muted. Game volume too loud. Model lagging. And more. Then please purchase Scuff Be Gone. Scuff Be Gone is the product you can spray in your face and never have to worry about scuff ever again. Scuff Be Gone's retail price varies from region to region and has an excessive price gouging addition. No guarantees are made of Scuff Be Gone's ability to actually remove scuff. Alright guys, we are back. So the good news is we're halfway through, which means 2 hours, so it's going to be standard stream length, roughly, give or take. Yeah, we got this, guys, we got this. We got this. God, just remember, this is just like when my kids have a double lesson. Except for, to be fair, if you, now's a good time for me to talk about that. If this was an in-class lesson, I obviously would not be so PowerPoint focused. It would obviously be a lot more participation based. And as the streams grow, if there are more people around, I will lean that way in stream as well. These are not designed to replace classroom teaching for anyone. The absolute best way is to have a great teacher who is engaging a whole class with activity and application. But I know that that's just not available to most people once you're an adult. You don't get to go back and have another chance. And so I'm hoping a little bit that I can make that happen for us. I can make it happen. Okay, let's go into some detail about this classification. If I'm going too fast, because I am going to pick up speed at places here, so if I'm going too fast at any point, please tell me to slow the F down, and I will. We're going to look at how we actually classify these organisms, okay? So we're going to start with something called the 3 domains of life. Now, you don't normally learn these immediately at school, but I've squished together 2 different levels. So in the 1990s, we changed how we were classifying things. We had RNA sequences from a bunch of organisms, from the ribosomes. We managed to reveal that actually life on Earth fell into 3 fundamental groups, above kingdoms. So this was above what we already knew, Animalia, Plantae, all of those. Above that, we introduced another layer called domains. Now, we don't generally learn these in lower school because they are newer. Okay, we had a 2-kingdom and 5-kingdom system, but this then added a 3-domain system at the top, which was much more specific, and it changed the 5-kingdom system as a result. We'll come to that in a moment. And so what we discovered is that everything falls genetically into 3 main groups, 3 main domains. Bacteria, these were single-celled prokaryotic organisms, and archaea, which were single-celled prokaryotic organisms that differed from bacteria. Okay? Now, the key difference here is before this, bacteria and archaea were always squished together as prokaryotes, and they were listed as a kingdom. Okay, we didn't have this level above, but we realized they were 2 separate things, and actually a lot of things fall beneath them, so they were separate. And then finally, everything else was eukarya. Eukaryotes are things that have a nucleus, membrane-bound organelles. So this is plants, animals, fungi, protists. The 4 things that we already recognize, hopefully, or maybe not protists, but we should recognize plants, animals, and fungi. The key thing here is that we used to have plants, animals, fungi, protists, and prokaryotes. Prokaryotes got removed and separated out into bacteria and archaea. These are 2 separate things. So we have these 3 domains of life, okay? And this is relatively new study. A lot of people don't realize, because it sounds like something old, right? These words sound like old words, but actually this is new. This is not something we've always known. So prokaryotes are single-celled organisms, and they don't have a membrane-bound nucleus. We do learn more about this in cells, but just to give us some context now. Their genetic material just floats around in the jelly of the cell, the cytoplasm. It's in like a region called a nucleoid, but there's no membrane holding it there. They have no membrane-bound organelles, in fact, at all. All of their organelles are just sitting there in the cytoplasm. They have really small ribosomes. Ribosomes are the organelles that read your DNA, essentially. We'll get to that in more detail later. They have a cell wall. Cell walls are usually made of cellulose or peptidoglycan. And they're also generally really small. They don't have to be, but they generally are. So why did we separate 2 separate domains? So they look really similar, okay? But actually they are about as different from each other as they both are from eukarya. So if eukarya is a separate group, they are a separate group to each other too. This is billions of years of them evolving separately. So archaea are actually closer related to eukarya than they are to bacteria. And that it's possible, as a result, that eukaryotic cells, things like humans, are evolved from a mutual ancestor with archaea. Just 2 seconds, guys. I just had a pretty urgent message. Oh no, really bad timing on their part, but I have to do it now because by the time we finish stream, they can't get it. 2 seconds, guys. I'm so sorry. Very unprofessional of me. Hashtag I'm fired. Oh no, I've lost an important file. Okay. Sorry, so yeah, we probably have a common ancestor there with the archaea that's much closer. Okay. And this was really important because we called this organization, the kingdom, phylum, all of those, we called them the tree of life, and this really changed the understanding. It's like it's a family tree of all living things, and it really changed how they worked. So what are the key differences between bacteria and archaea that made us think this? So they're both prokaryotes, so they both have those same things, no membrane-bound organelles, smaller, etc. But the lipids, the fats, okay, they have different types of fats. Okay, so in a bacteria, the membranes have glycerol-based fats. This is the same as we have, okay, in our membranes. But archaea cell membranes have a different type of fat, okay? Their chains are different. They don't have the fatty acids. They have isopropanines. They still have glycerol and ether bonds. They have ether bonds, but they don't have ester bonds and fatty acids. And this makes them more stable in extreme conditions. So this means that archaea can actually generally support more extreme conditions than bacteria. For ribosomal RNA, okay, bacteria have really distinctive ribosomal RNA, whereas archaea's ribosomal RNA is very similar to us, and that's what suggests a closer evolutionary relationship. DNA stuff being roughly the same is a big sign that you're very similar to us. And then the cell walls are different. Peptidoglycan is bacteria, we know this, we're confident with this, but archaea don't actually have that. They have lots of other types of cell wall, and the cell walls are pretty fixed in bacteria what they are. So the domain eukarya, this is the one we're all most familiar with. This is the one you'll learn in like elementary school, right? You don't learn eukarya, but you do learn the things in it. So these are all the things that have true nucleus cells. They're all very similar to us. We have lots and lots of them. The key ones here are, that you might not know, are Protoctista. You should know Animalia, animals, Plantae, plants, fungi, but Protoctista might be new to you, okay? The protists might be new. And with the domain eukarya, we then continue down this classification. Now, to be clear, bacteria and archaea have their own hierarchies. They don't follow this taxonomic hierarchy. Okay, they don't follow this taxonomic hierarchy. They they have their own. But this is the one that eukaryotes, plants, animals, everyone follows this, which is that you have the kingdom, the phylum, the class, the order, the family, the genus, the species. And the ones you'll be most familiar with are the animal kingdoms, probably. Most of you will have learnt the animal kingdoms growing up. Okay. So remembering the hierarchy. Okay. We normally use mnemonics. Okay. Goes domain, kingdom, phylum, class, order, down. Okay, so domain, eukarya, kingdom, animalia, phylum, chordata, class, mammalia, order, primates. Okay, as you go down, as you go down, as you go down. And the classes you've learned probably come from the kingdom, animalia. You probably learned mammals, reptiles, those. Most of those will come from the kingdom animalia that you've learned. You might have learned some plant ones depending on how high you studied science. Okay. So, kingdom is a level of classification in our order. So we can split things into domain, bacteria, archaea, eukarya. Then within eukarya, we can break things down again. So we can go down from the eukarya to kingdom, animal kingdom, plant kingdom, for example. What makes a plant a plant? What makes an animal an animal? And we can continue to break that down as we go through. So what does make an animal an animal? So the animal kingdom are multicellular organisms. They have no cell wall or chloroplasts. Chloroplasts are the cell object that does photosynthesis. Most animals can move location, and most animals ingest solid food and digest it internally. Okay? They digest inside their body. In the animal kingdom, there is an incredible variety. Okay, remember, animal kingdom includes things like birds. People often forget. So how do we describe that using more scientific wording? So we say they do heterotrophic nutrition. Heterotrophic. So trophic is a word used about food and energy. So plants are autotrophic. They create their own food. Heterotrophic means that they need to get their food from something else. Okay, so they need to get it from something else. They need to ingest it and digest it. They have no rigid cell wall at all in kingdom Animalia, just a cell membrane, okay, for more flexibility in their cells. They're multicellular. That means they have more than one cell, and those cells will be specialized into specific types for different jobs within their body. And their movement and senses are the most extreme. They can move their entire body from place to place. And they have multiple sense organs, so they can respond rapidly to their environment. There's some other little things that go above this level of knowledge. So for example, we mostly know that they develop from a blastular stage. They mostly reproduce sexually, although we're discovering some asexual reproduction in animals, which is interesting. They store carbohydrates as glycogen. That's a pretty standard thing. And they can include invertebrates and vertebrates. So that's actually something that's sort of a difference, okay? Now the plant kingdom, they're also multicellular, but they do have an outside wall, and they do have chloroplasts so that they can make their own food with photosynthesis. So that's the key differences. Autotrophic nutrition, they make their own food using photosynthesis. They have cell walls made of cellulose, and they are multicellular still, so that's a similarity. Okay, so those chloroplasts are important. They don't have excretory organs. They do excretion, but not through specific organs. They don't need a nervous system, for example, for their senses. Their senses don't rely on that. And they have a pretty huge amount of diversity, just like the animal kingdom. We just don't spot the diversity. But mosses and ferns are very different to flowering plants, for example. Trees are very different to a moss. And then we break down these features. So the animal kingdom has chordata, which are vertebrates, and those vertebrates, those chordata, break down into classes: mammals, birds, reptiles, amphibians, fish. we should already be really familiar with these. Mammals are the ones that have usually 4 limbs. Doesn't have to be arms, doesn't have to be feet, but they're 4 limbs. Warm-blooded, hair or fur, they have a diaphragm. So that's, if you look in the diagram here, the diaphragm is this muscle under your lungs that helps breathing happen, helps push, expel out gases and pull in gases. They produce milk and they give birth to fully formed young. There are some exceptions. We'll talk about that later. Fish are cold-blooded. That means that their body doesn't control its own temperature. Its temperature is controlled by its environment. It has overlapping scales. The scales of fish are actually incredibly cool. Like incredibly, incredibly cool. They have fins, okay, to help them swim. They have something called filamentous gills. So this is gills made of filaments that can basically, instead of our breathing and our diaphragm, they get their oxygen by filtering water through it. And eggs are fertilized external to the body, because there is a difference between an internally fertilized egg and an externally fertilized egg. So in fish, they lay all their eggs and then the sperm is laid across the eggs, externally fertilizing them. Look at them gills. How cool does that look? To me, that's insanely cool. Maybe not to you, but to me, that is wildly cool. Okay, reptiles are cold-blooded. They live on the land and they have really dry, scaly skin. They lay eggs and their eggs have a really tough, rubbery shell, okay? So they will often bask in the sun to raise up. Even the ones that go in the water are land living, okay? So things like alligators are land living, but they go into the water. They can swim, they can stay underwater a long time, but they are land living. This is also a confusion, is often that reptiles have scales, and some of them do have limited scales, but actually for the most part they have like a scaly skin. It's not these interlocking scales like fish have, it's skin that kind of has a scaly texture. And their eggs are much tougher than fish's, much, much tougher. Birds are warm-blooded, just like our mammalia. They have fused vertebrae though, okay? Their vertebrae are fused together. They don't have the movement in their backbone that mammalia have. They have feathers, usually. They have 2 wings, 2 legs, and they lay eggs with very hard shells, okay? Even harder than reptiles. They usually have some form of claw, and they have beaks. When we say they have some form of claw, it might be almost a vestigial claw, like we have a vestigial tail, like a tail that isn't really a tail anymore. It may actually happen. Okay. Birds are pretty incredible. Okay. And then we have amphibians. These are cold-blooded again. So the only 2 warm-blooded examples we have here are birds and mammals. Okay. Amphibians are cold-blooded again, 4 limbs. Moist skin again. Amphibians have skin, reptiles have scales, scaly skin. Amphibians have like moist skin, no scales even visible. They are carnivorous, almost universally. I've had to think universally. They live on land and in water. They are comfortable living in both. This is a key difference between a reptile and an amphibian, is an amphibian will live part or all of its life in water. And their eggs are like jelly-covered eggs, okay? And they're fertilized externally, similar to how fish are, okay? So water-living organisms have these externally fertilized eggs, and amphibians, being water-living, do that. Look at that newt. I love newts, they're so gorgeous. Just so cute, just so cute. Also tell me that these amphibian eggs don't look like tapioca. Tell me that's not. That's tapioca right there. You can tell me that's frog spawn all you want. To me, that's the beginning of a boba. Amphibian egg tea. Delicious. Okay, so that is our vertebrates. But then we have our invertebrates, our arthropods, and they also have separate groups within them, classes. So remember, our vertebrate, and then below that we have a class. So we have kingdom, phylum, class, I think. And then the class here would be insects, arachnids, crustaceans, and myriapods, all separate to each other. And invertebrates is where most people get it wrong, because we don't generally look at them in so much detail in school. So unless you went on to further study, you won't have looked at them, and we don't like to think about them. They're not really the members of our eukaryota who we care about, if we're being honest, right? So for arthropods, if they're an insect, they have a segmented body, head, thorax, and abdomen. I am pausing long enough on each screen that if you're reviewing this, you'll be able to go back and pause if you're preparing for an exam. They have 3 pairs of jointed legs. They have compound eyes. That's eyes that have one eye but multiple kind of lenses within it. 1 pair of antennae. A cuticle, which means it's got a hard exoskeleton, and they often have wings, but they're not required to. So beetles, ants, wasps, bees, flies, butterflies, moths, these are all insects because they have these segmented bodies and legs and the compound eyes. Okay? And you can see the similarities, but you can also see they can look very, very different, but they still have the segmented body, the segmented legs, the compound eyes, and the antennae. Okay, they have the same thing. When insects do have wings, remember they don't have to, but when they do, they mostly have 2 pairs. They do mostly have 2 pairs. Your arachnids, I think this is the group people are most aware of. They have 8 legs. Their head and thorax are combined, and they have several pairs of simple eyes. So they don't have the compound eyes with multiple lenses, but they do have multiple eyes, and they have no antennae, no wings, okay? Pedipalps are things that are used to bite, poison, or immobilize prey. They're your spider fangs. Your spider fangs are the pedipalps. They're basically pedi, feet. Palp is as in palpation, okay? So they are feet that allow them to do something, Crustaceans have 2 pairs of antennae. Look at that, you can see here very clearly. They have a little one and a big one. They have compound eyes like our insects, and they have a protective exoskeleton like our insects. But they have 5 pairs of limbs, and I don't think people often realize crustaceans do all have 5 pairs of limbs. And just like our arachnids, they actually have a combined head and thorax. So if you look at arachnids and you look at insects, somewhere in between the two like behaviors, you get crustaceans. And we mostly know crustaceans for our deliciousness. But look here. Look at this poor delicious fella. This is a woodlouse. And he's in the same family as these. People often think a woodlouse is an insect, but a woodlouse is a crustacean. He has 2 pairs of antennae, compound eyes, a protective exoskeleton, 5 pairs of limbs, 1, 2, 3, 4, 5, and a combined head and thorax. It's not an insect. Not an insect, see? Wow, look at these. It's crazy detailed pictures. And then lastly is myriapods. These have a heavily segmented body with a pair of legs on each body segment, but they can have a bunch of different segments, at least 10 though. So at least 10 segments, but they can have way more than 10 segments too. And they have 1 pair of antennae and just simple eyes. They're pretty simple other than being heavily segmented. And we'll know these as centipedes, millipedes, the varying peds. And they're called myriapods. Myria like myriad, like many, and pods feet. They're literally the many feets. We all love a good many feet. And then again, I mentioned that plants have their own breakdowns too. So we have ferns, and then separately we have flowering plants. So ferns have stems below the ground. They do not produce seeds. They produce single-celled spores, okay? Much like fungi do. They release these spores on the underside of their leaves. It makes them very different from flowering plants. And you can actually see the spores on a fern at the right times of year if you go up to one. And then flowering plants reproduce by seeds that are formed in flowers. And within that, we have monocotyledons. These are ones that have one embryonic leaf inside their seeds, okay? And their flower parts are grouped in threes, and they'll have these like long, narrow leaves with parallel veins. Long, narrow leaves with parallel veins, like you can see here, going up and down them. So here, dicotyledons. So these are two embryonic leaves inside their seeds, and their flowering parts are grouped in fives. That doesn't mean they have five, just like the last ones doesn't mean they have three. They could have 6 in the last one. They could have 10 in this one. It's like the multiples they have. And they have broad leaves with these branching veins. The last ones were thin, straight leaves with these going. So kingdom is a level of classification. So you have animal and plant kingdoms, and then within that, as I mentioned, you have the phylums. Fern is an example. You have the phylums, and then you also have the classes as well. So there's a huge amount of breakdown. So the phylum for animal is chordata, vertebrates, and arthropods. Okay, so chordata and arthropods. For plants, it's ferns and flowering plants, and then within flowering plants, you have classes. Monocotyledons, dicotyledons. It just keeps going down like a tree, like a family tree. But I did mention that there are 3 other main kingdoms: fungi, prokaryote, and protoctis. Prokaryote, I hear you say. We will talk about that. So fungi don't have cells, kind of. They have something called hyphae. Hyphae work the same way as a cell, but they are long, thread-like, and they form structures that we call mycelium. And if you've been on TikTok recently, you'll have seen mycelium everywhere. There's a guy showing his mycelium cultures growing in his thing. And the nuclei are just scattered out throughout the cytoplasm in those hyphae. There's no distinct this cell, this cell, this cell. And they release enzymes, and they digest the food outside of their body, okay? Not within their body. They digest it outside their body, and then absorb the nutrients through their cell walls. They just absorb just the nutrients then in, okay? And we're really used to seeing things like mushrooms, but we're not so used to seeing the hyphae of simpler mycelium-based organisms. Okay. So mold is also a fungi. So mold breaks down your food to absorb the nutrients. And the reason that moldy food is a problem is the chemicals they let out to digest the food can harm you. That's why usually if you're eating the non-moldy part of a food that's got mold on it, you'll be fine, because they haven't done anything to that part. So this is, for us, mushrooms, molds, yeasts. We used to call them plants. So sometimes people will get confused and say, aren't they plants? And that's because we did used to classify them as plants, but we know they're different now. We know there's too many differences to call them plants. They have a eukaryotic cell with a true nucleus. They have a different type of cell wall to plants. They do heterotrophic nutrition. They can't photosynthesize like a plant can. They do something called saprotrophic feeding. So saprotrophic feeding is when you excrete the enzymes onto food, then absorb it. So it's still heterotrophic nutrition. You're getting it from outside of you, but they're getting it by digesting it outside their body. Okay. They still store as glycogen like an animal does, not as starch. Okay. And they produce spores to reproduce. And they can reproduce sexually and asexually. People sometimes get a bit confused by that. Prokaryotes are redundant in the modern domain system, okay? But examples were bacteria and blue and green algae. They're single-celled organisms. They don't have a nucleus, okay? We don't need to know much more about them because they don't exist as a kingdom anymore. But before we had the domain system, they did. And so a lot of people still refer to them in the kingdoms, and it's useful to know that you're encountering that. Protoctists are single-celled organisms. Their chromosomes, though, have a nucleus, which makes them unusual compared to bacteria and archaea. And some of them can actually have chloroplasts to make their own food. Some of them digest their food internally. Okay. They're probably one of the more interesting. They are very loosely defined. Basically, everything that doesn't fit into fungi, plantae, or animalia and is eukaryotic, we just kind of put it in here. And there's a lot of debate as to whether we need to split it because it's got amoebas in it. They're heterotrophic amoebas. It's got paramecium. Okay? It's got chlorella, who are autotrophic. These are all super different to each other, and there's a lot of talk that maybe we should split them up. Maybe it's time to make it even more complicated again. Okay? Okay. It's been a lot of me talking. So I have some organisms here. Can you have a think about what you think they belong to in terms of the classification? So here we're looking for, are they Protoctista, fungi, plantae, or animalia? I'm going to give you, you might not get through all of them, but I'll give you 3 minutes. All right, how did we do? Multicellular organism found in a fallen log in a forest. No chlorophyll, releases spores from its mushroom-shaped fruiting body, and its cell walls are made of chitin. What do we think this one is? It is, drumroll. Fungi! Organism B is a single-celled organism found in pond water. It has chloroplasts, makes its own food through photosynthesis. It has a cell wall and flagellum for movement. Which one is this? Drumroll, it is. This one is Protoctista. It does do photosynthesis like plants would, but it's a single-celled organism. That means not got multiple cells. And it has a cell wall, and it has a flagellum. So that's going to put it into Protoctista. Plants don't have flagella. Okay. Number 3, organism C, a multicellular organism with roots, stems, and leaves. Its cells contain chloroplasts and have cellulose cell walls. It produces flowers and seeds. Let's see. Drum roll. This is the world's worst drum roll because I've got a space. It is... It is Plante. Woo. Organism D is a multicellular organism that lives in the ocean. It has specialized organisms including gills for gas exchange and fins for swimming. Its cells have no cell walls. It is Animalia. Organism E is a single-celled organism that moves using tiny hairs called cilia. It feeds by engulfing bacteria. It has a nucleus, but no cell wall. What would organism E be? Yeah, I would say it's Protoctista. And what about 6? A multicellular organism growing on the side of a tree trunk. It's green and photosynthetic, but lacks true roots, stems, or leaves. The structure is a flat thallus. Ah, yes. It can't be fungi because it's photosynthetic. Fungi are not photosynthetic. Okay, that's always one of the key ones is to use photosynthesis to knock out some of the categories. Okay, let's move on. Okay, so here, and I'm going to leave this up for a moment to let people have a look through. If you are reviewing or going over the content, this is a slide I recommend you pause to take a look at. This is the kind of breakdown of what you're looking for. Okay, this is what you're looking for, whether they're multicellular, unicellular. Okay, so plants, if it's unicellular, it can't be a plant or an animal. Okay, that is some key points. And if it does photosynthesis, it has to be Plantae or Protoctista. Remember, moving, all of them can move. It's whether they can physically move their whole body from one location to another. That is the difference. Okay. So what about viruses? Because this is always the question people have. Are viruses alive? Are they alive? And we can look at this scientifically. They are a unique challenge. It would be wrong to claim they are not a unique challenge. They're not included in any of the domains or kingdoms that we've discussed. They lack many characteristics we associate with life. They actually don't meet Mrs. Graham. They can't reproduce independently, carry out metabolism, or respond to stimuli on their own. They could only function inside our cells. And so this has no simple answer. They exist in this really grey area as to whether them needing to exist in our cells is simply parasitic or whether it is an example of not being able to reproduce. Outside of a host, they are just particles. Inside of a host, they hijack the machinery to reproduce. And how we understand how they're related is helping us predict their behavior. We can develop treatments, we can track disease, we can create vaccines, we could create the equivalent of antibiotics, antivirals for them. It's really important we study their classification, but they don't fit into this classical form of classification. They all have dRNA or RNA surrounded by a protein coat that we call a capsid. Some viruses have like outer lipid envelope too, and they are incredibly small, far, far, far smaller than bacteria. This makes them fascinating to fight because there really isn't anything to fight. There's very little to a virus other than what it takes from the host. The same things that make cancer hard to treat because it's just our cells make viruses hard to treat because they're just using our cells. They're not part of a kingdom though. We don't demonstrate these characteristics. They don't eat, respire, excrete, or grow. They have no nucleus, cytoplasm, cell organelle, or cell membrane, and they reproduce only within the context of the cells of other living organisms. So we don't classify these using domain, kingdom, phylum. Instead, we classify based on the type and structure of their genetic material. It's called the Baltimore classification system, developed by David Baltimore, and it's the most common approach. So number one, the type of nucleic acid. So does it have DNA or RNA as its genetic material? This will then decide how it replicates inside our cells, so it's really important for us for treatment and prevention. Is it single or double-stranded? Is the nucleic acid single-stranded, one strand, or double-stranded, two complementary strands? This affects how it replicates. And additional features like capsid shape, presence of an envelope, host range, whether the viruses are positive or negative sense of their RNA. We can talk more about that when we go into DNA and RNA. And so we end up with these kind of categories. We have double-stranded DNA viruses and single-stranded DNA viruses. We have double-stranded RNA viruses. We have single-stranded RNA viruses. So coronavirus, influenza, HIV, polio, measles are all single-stranded RNA. Okay? Some will replicate directly and others just have to make a complementary strand. Then double-stranded RNA viruses replicate using viral RNA polymerase. Okay? So RNA viruses use a little bit more of their own process, I guess, than DNA viruses. RNA viruses are able to mutate more rapidly. That's because the RNA replication is less accurate, so more mistakes are made, and that's what causes mutations. This is why you have a new flu vaccine every year. It's why during COVID, it was so hard to cover all the strains. You remember we were having new strains come out like every couple of weeks? Some of those new strains were fairly irrelevant and some were quite significant, and that is because that RNA replication is so inaccurate that mutations were happening every few generations. And we were having these new strains develop that were just, it was like playing whack-a-mole to try and keep on top of. Every year as well with the flu vaccines, the vaccine you're getting is a predicted vaccine. They're predicting which strains they think will be prevalent that season and including those strains in your vaccine. That's why some years the flu vaccine doesn't work very well. Loads of people get flu if there's been some sudden change or because of how quickly they mutate and new mutations taking grip after the predictions were made. Most of the time our prediction models are really good for it and the flu vaccine is highly effective, but very occasionally you get a year where unfortunately the viruses are a step ahead of us and the flu vaccine is less effective. This does not mean flu vaccines should not be got. This does not mean that vaccines are ineffective. It means that we as humans are just slower at adapting than a virus is, and there's only so much we can do about that. So here we go. Some of you will have seen the classic COVID-19 models that were going around, the footballs with the little sticks on. HIV is very, very, very similar actually, really. Okay, except is so it's a single-stranded RNA. It's what we call positive sense RNA. We'll go into more details when we get to DNA and RNA. It's enveloped, uses reverse transcriptase. Okay, the key point here is that HIV actually converts RNA to DNA inside our cells, and its host is our immune cells. So that's why HIV tanks your immune system. It is targeting and using up your immune cells, so you are unable to. Kind of crazy. And why does this matter? As we've said, for vaccine development, antiviral treatments, for tracking outbreaks, for predicting where it's going to go and how it might spread. COVID-19, this was especially important. Understanding evolution. Being able to see how new diseases might emerge, or which new diseases are at risk at jumping from animals to humans, can help us get ahead of viruses. And the more ahead of a virus we are, the better, because they adapt quicker than we do. Okay. We're not going to do this, but if you're someone doing this as a review practice, you do need to do some. So if you're someone doing this as review practice, once you've gone back, reviewed your content, please pause this slide and have a go at classifying a domestic cat or another animal of your choice using the system that we've done. See if you can find out its domain, phylum, order, genus, kingdom, class, all of that. I've put up the example for Felis catus, but you should pick an animal and you should see if you can classify it using this same logic. Okay, so if that's going to be you for now, now's the time to pause. And for the rest of you, let's carry on. Okay, we're not doing this one either. I just forgot to remove it, my bad. Okay, it is true or false. There are 10 questions here, so I'm going to give you 2 minutes to go through and just give yourself a quick tour of false notes so you can go through and check your answers in a moment. All right. Okay. True or false? Let's go through. Number 1. Oh, wait, these are coming up in a crazy order. I'll just pop them up. There we go. Viruses are not living organisms by the definition we learn at this point. Eukaryotic cells do have a nucleus. Fungi do store their carbohydrates. Glycogen. All plants... Glycogen. All plants photosynthesize. Bacterial cell walls are made of cellulose. Is incorrect. Peptidoglycans. Chlorella is an example of a protoctist. Yes, there are 5 kingdoms. I've said yes, but how many are there under the modern classification? Four. Yeah. A lot of curriculums still teach 5 though, so it's really important to be aware of if you are someone sitting exams. Protoctists have plasmids. No, plasmids are circles of DNA in bacteria kept outside of the main DNA. Plants store their carbohydrate as starch, and all fungi are single-celled. We're going to dive more into DNA, RNA, and different types of cells in later topics, so don't worry if that was all a bit new to you. Again, for those of you doing some study and need some practice at home, I'm going to ask in a moment that you pause here, and some answers will come up when you restart the video. So if you're someone who needs the study practice, you can pause now. And these are the answers to your study practice. Okay. So we're nearly at the end of today's topic. There's some challenges for classification, and it's always worth being aware of what this means in the real world. This is a really cool virus switch, by the way. Some of the challenges are things called convergent evolution. So sometimes unrelated animals will get similar features because the environmental pressure is the same that they've experienced. So for example, dolphins are a mammal and sharks are fish, but they do look similar because they live in a similar environment and have the same pressures put on them, which means that certain features have been desirable for both of them. And this can make morphological classification misleading if we're not really careful. And sometimes DNA evidence contradicts physical appearance. So genetic analysis has led to the reclassification of a lot of organisms. So for example, we looked at the DNA of hippos, and they're actually more closely related to whales than to other hoofed animals. So we needed to reclassify them. And then there's some populations where they interbreed with their neighbors, but not with distant populations of the same species. So this challenges what we call the biological species concept and shows that it can be fuzzy. So we teach you this very black and white now to teach you a rule that you can understand. But when we get to, there are exceptions and there are complex cases. Okay? And it's almost impossible to teach someone in one hit all of the possible things that could happen. We have to build that knowledge over time. One of the key points of scientific study to be aware is there is probably someone who knows more than you about a particular topic, and to respect and learn from that. And then there's horizontal gene transfer. So for bacteria and archaea specifically, they can actually transfer genes manually between species, so not via reproduction or genetic behavior. They can literally inject DNA into each other, and that can make it really hard to use genetic similarities for them, because an individual you're testing may have had a little injection of a gene from someone else. So modern classification is using more and more what we call phylogenetics. So that's where we look at the evolutionary relationships. So we don't just base shared characteristics, we base it on their evolutionary history and common ancestry, which means we need to know their DNA. Okay? So cladistics is when we take those shared characteristics, okay? And a cladogram is a diagram showing it. Then we use molecular evidence. We use DNA. We use mitochondrial DNA. Mitochondrial DNA is so useful for evolution. And we use whole genome sequencing, and we get a complete picture. So the traditional approach, we just looked and we made subjective decisions. In the modern approach, we combine all of this knowledge to create an evolutionary relationship. And it has meant we have reclassified a lot of organisms. For example, birds are now classified within Dinosauria because they're descended from dinosaurs. The 3-domain system itself is due to phylogenetic analysis. So this idea that we've learned some basic ways, and that's certainly how the journey started, but the more methods we have to analyze an animal in depth or a plant in depth, the more we're able to place it. But why keep improving it? Why do we need to? Conservation. Classification helps us know which species are most in need of protection. If you're a unique evolutionary lineage, conserving you becomes a priority to maintain biodiversity and genetic diversity. If you are part of a complex tree of 1 million species very similar to you, you are probably less important in terms of biodiversity and genetic diversity in order for us to do. Medical research. Knowing how organisms are related helps us predict where diseases might jump to. Okay, it also helps us in terms of deciding what medicines we can get from different things. Agriculture, food security. So picking disease-resistant strains or crops we could use that are more disease-resistant, both in terms of growing that crop, but also in terms of taking a gene from that crop and putting it into another, like our golden rice with vitamin A. Managing ecosystems. Try to work out which species are truly invasive, i.e. humans have caused the problem, versus naturally invasive, they've just grown and developed, so that we aren't involving ourselves in an ecosystem we shouldn't be. Discovering new species. There are so many species, literally millions of species we think we haven't discovered yet. So being able to classify them is an essential way to find them and work out what they are, because the species we've discovered already are the very visibly big, obvious ones. We're talking different arthropods. We're talking things with very small genetic differences that we have yet to discover. And because we can now look at the relationships of different species, we can see how they've changed over billions of years, which means we can see how different branches of the tree have adapted to different environmental change, which means we can predict how species might. For the record, humans are likely to die out with significant environmental change. We are one of the species most affected by the environmental change we are causing. Okay. Just for those people who are studying at home, here's a little summary of what we've done today. This is a lot of stuff. We are going to dive into some of the concepts like DNA and cells in more detail moving forward, but we've covered a lot of stuff in one day, like a lot of stuff. You guys have done great. And for those of you who find the language difficult, these are some key words you should make sure you go away and you are comfortable with if you want to be able to talk more articulately about this topic. And I'm just going to let you know that this is your final little section to test your knowledge. So if you are someone studying at home, now's your chance to pause your screen. And here are your answers to go through and check your work. Again, you might want to pause your screen. And here is part 2. If you need to check them properly, just pause your screen one last time. And topics you might find interesting to research that don't really fall within most school curricula but are the next steps to learn about. Whole genome sequencing we will get onto. But you might also like metagenomics, okay? These are really, really, really cool. Artificial intelligence and how that can be used, machine learning, to analyze these data sets to find these patterns. DNA barcoding, okay, to be able to identify species really quickly. Another thing you might find interesting if you've enjoyed this topic is epigenetics. Epigenetics is things that affect us from our environment that we then can pass on genetically to our offspring. Okay, and here is just a little task you can give yourself if you wish to, those of you studying at home. It's a 5-minute task, so I would pause, give yourself 5 minutes or so before you restart. And you have officially managed to cover a significant amount of information. In the end, we will conserve only what we love, we will love only what we understand, and we will understand only what we are taught. We are seeing this in the world today. People do not understand fundamental scientific concepts, so they don't care about them and do not value their importance. And I think it's worth taking a moment to realise how we've ended up in a world like we have. Thank you very much for joining me. Hopefully, if today's topic was a bit dry for you, you'll find the next topic's a bit more involved, but it is really important we build the foundations of the knowledge to get to where we're going to go. Thank you for joining me in Britesparks Hagwon, and I'll see you tomorrow for Fran Bow. Woohoo!